Hi everyone, this video is about exocytosis. So we are talking about endocytosis and exocytosis. Endocytosis is the process of bringing things into the cell. Exocytosis is bringing them out of the cell. And so what does the cell need to secrete? Well, if you think about the way this works, it's a vesicle. And a vesicle is made of lipids. And so when a cell exocytoses, vesicles will join with the lipid membrane. And so this is a way to actually bring lipids to the cell surface. This is how it's going to bring membrane-bound proteins, channels, transporters, all those things we've been talking about. How do they get to the outside membrane? How do they get to the membranes inside the cell? Process of exocytosis. Digestive enzymes. This is something that on an organismal level, cells make digestive enzymes, and they secrete them through exocytosis. Extracellular matrix material, right? The things that kind of give cell structure in the outside of their environment. This is secreted by the cell. And so there are two types of exocytosis. There is constitutive secretion. So this is just happening constantly. It is unregulated. This is often for things that need to be secreted, like a digestive enzyme or something like that. And then there is regulated. Regulated exocytosis only happens when there is a signal present. Right? So as you can see here, an extracellular might come in, hormone, neurotransmitter, and that might signal exocytosis. Constitutive is also how waste is gotten rid of in a cell. And so what is secreted? In constitutive, this is how lipids get to the membrane to expand the membrane. Matrix proteins, signaling molecules. This is present in all types of cells. Regulated exocytosis, this one that can be triggered by a signal, is usually only present in very specified cells. And the density of what can be excreted in regulated exocytosis is much greater. So things like neurotransmitters and hormones are done here because everything is actually specifically packed into a vesicle to be released. When this happens, what you want to think about is that vesicles have orientation. And so when you look at this diagram, look at the Golgi, right, which is where these vesicles are coming from. And then notice that membrane protein, right, its hexagonal head is inside the Golgi. When it becomes a vesicle, that hexagonal head is inside the vesicle. When it joins with the plasma membrane, that hexagonal head is now outside of the cell. So notice that that head on that molecule never touched the cytoplasm. And so this is a way that depending upon how these proteins are put into the Golgi membrane, it can be dictated which side will be extracellular and which side will be intracellular facing in the cytosolic and non-cytosolic phase of the membranes. Right, so again, this is also how lipids are put in, but they remain constant, and they also can be put in very specifically. So I mentioned that vesicles can dock and fuse with the membrane. So this is a very rough schematic of what that looks like. It comes up, the membranes merge, and then become just a flat membrane. Now, this is not just happening on its own. What happens is that those two membranes would never actually get quite close enough or be pushed hard enough to fuse into one on their own. And so you have protein interactions that are mediating this process, right? And what I want you to pay attention to are these molecules called snares and our tethering proteins. So V-snares are on the vesicles, and they are going to bind to the T-snares, and they actually kind of pull the vesicle close like this so it can fuse with the membrane, right? And so this is a protein-mediated vesicle fusion. The protein called Rab on there, which is what's recognized by a tethering protein. So again, this is a theme that you're going to start to see over and over again as we start to talk about the specificity and very specific interactions in the cell, is that it has to be recognized, and then there's usually action happening. And so secretion of a signaling molecule, the classic example of secretion is in the neuron. And so what I want to show you here is this picture of synaptic vesicles, right? So they're vesicles that hang out where the neurons talk to each other. And you can see them there, and there is an actual image of an exocytosing vesicle. This is also used by amoebas, right? So amoebas are single-cell organisms. So to bring in food, they bring in food using phagocytosis, which is discussed in the endocytosis video. They digest it, and then they have waste. Material they're not going to use. That is then expelled using exocytosis. It's often easier to think about this in terms of a single-celled organism that you know brings in material, uses some, and then gets rid of old material it's not using. That's waste. Organisms produce waste too, but all that waste comes from cells excreting their waste, and that gets packaged by the organism. Right? So again, sometimes it's easier to think about this on a single cell scale if you think of this as a whole organism, but your cells do this too. And so that's all we're going to talk about for exocytosis. And so I'll see you all in the next video.